Hello, today's session at Muhumuza Naphtali medicalsciences.com, we want to talk about electron transport chain under biochemistry. Previously, we have seen glucose metabolism. That is, we saw glycolysis, how we convert glucose, a 6-carbon sugar, into a 3-carbon sugar. We saw Krebs cycle, or what we call tricarboxylic cycle. So today, lastly, we want to look at electron transport chain, which is the end. So the last products of metabolism is fed into the electron transport chain. So that is what we want to talk about. Or sometimes we call it oxidative phosphorylation. Let's get started. So electron transport chain, as we have seen, it is the final stage whereby products of glycolysis and Krebs cycle are used. And in this process, we see the end products majorly we feed in electron transport chain is NADH and FADH2. These are the products from glycolysis and the Krebs cycle. Whereby in glycolysis, as we saw, that in glycolysis, you produce 2 NADH. Then in the transition stage, we also produce other 2 NADH. Then we see in the Krebs cycle. In the Krebs cycle, we are producing 6 NADH. 2 molecules of acetyl-CoA, we are producing 6 NADH plus 2 molecules of FADH2. So these are the products of carbohydrate metabolism under glycolysis, under transition state, and under Krebs cycle. The first 2 under glycolysis and transition step, they do not directly enter into the electron transport chain. They are shuttled because they are outside the mitochondria, and whereby this electron transport chain is occurring into the inner mitochondrial membrane, along with the inner mitochondrial membrane. So we shall see the shuttles that make these 2 NADs to enter into the mitochondria. So if I can illustrate here, is that this electron transport chain being the final acceptor or the final step to produce ATP, we see it occurs along the inner mitochondrial membrane. It occurs along the inner mitochondrial membrane. That's why it occurs. It is not in the matrix, but along the inner mitochondrial membrane. We know that mitochondria is made up of cristae, whereby the cristae, this one we call it the inner mitochondrial membrane, then it has the outer mitochondrial membrane. So we see this electron transport chain occurring along the inner mitochondrial membrane. That's where it occurs. So I'm going to enlarge this, and I draw it here, whereby this one is going to be the inner mitochondrial membrane. This is the inner mitochondrial membrane. We're calling it inner mitochondrial membrane. That is where it occurs. Whereby this one is the intermembrane space. This one we are going to call it intermembrane space. Intermembrane space. This is the space we see when we have this inner mitochondrial membrane. The space between the outer and the inner is what we are calling the intermembrane space. That is this part. Then this one is going to be the matrix. This side is going to be the matrix, the mitochondrial matrix. And we see different complexes doing the role of electron transport along the inner mitochondrial membrane, whereby we have complex 1. We have complex 1. This is complex 1, we have complex 2. This is complex 2. We have complex 3. This is complex 3. We have complex 4. This is complex 4. And we have complex 5. And this complex 5 shall see its law. This is complex 5, which we call ATP synthase. ATP synthase. So these complexes are along the inner mitochondrial matrix. So we want to see the role of each complex along the inner mitochondrial membrane. So we see complex 1, its other name is called NADH. NADH, coenzyme 2 reductase, whereby it is characterized by the NAD dehydrogenase. Then complex 2 is also known as succinate, coenzyme 2 reductase. Then this one is known as complex 3. Complex 3, we see it as being cytochrome C. This one, it involves, we shall see cytochrome C playing a role here. Cytochrome C. Then we have here, we have the coenzyme 2. Coenzyme 2 doing the role. Then this is cytochrome oxidase. Cytochrome oxidase. So we want to see how electrons, as the name suggests that it is electron transport chain, we want to see how electrons move from one compound to the other. And this reaction involves oxidation, reduction reaction, what we call redox reaction. That if we have NADH, that this NADH, we see it giving up its electrons to form NAD plus. And of course, when we lose electrons, that process is known as what we call, it becomes oxidized. Loss of electron is oxidation. It becomes oxidized, and this one occurs in presence of NAD dehydrogenase enzyme. So this electron, which is given up into the complex 1, it undergoes redox reaction. One compound, for example, compound like flavin mononucleotide and sulfur, iron sulfur. These are the two compounds that are acting as electron carriers. So we are going to see flavin mononucleotide, iron sulfur, and some cytochrome C, cytochrome A being electron carriers. That when NADH donates its electrons, it undergoes oxidation and reduction. And this oxidation and reduction will cause some cleaved hydrogen ions in the mitochondrial space to be moved outside into the intermembrane space. And before we continue, we have to note that this inner mitochondrial membrane is impermeable to protons. Protons only pass through complex 1, complex 3, and complex 4. Complex 1, 3, and 4 are the only complexes that are permeable to hydrogen ions. But complex 2 is not permeable to hydrogen ions. And these hydrogen ions, which are produced by oxidation reaction, do not pass along the inner membrane space. They only pass through complex 1, 3, and 4. So whenever NAD is oxidized by this dehydrogenase enzyme, the hydrogen or the proton is produced, pass through the complex 1, and they go into the intermembrane space. Then the third, which is the product of Krebs cycle, for it bypasses complex 1. It goes directly to complex 2, which we call succinate coenzyme 2 reductase. So we are going to see that these electrons which are produced in the complex 1 are passed over by coenzyme 2. They are given to coenzyme 2, and coenzyme 2 pass them over to complex 2. These are the electrons. And these electrons excite complex 2, whereby it will come and oxidize the FAD. That when we have FAD-H, if I can rub this word, the FAD-H which is formed is going to be oxidized by complex 2 to oxidize the FAD, with the release, whereby this FAD is going to donate electrons also with the release of protons. We see protons being released. But remember, complex 2 is impermeable to protons. So we don't see exit of these protons via complex 2. So these electrons continue to undergo redox reactions, and these redox reactions, after this one becoming excited, it passes its electrons to complex 3. So we see the electrons being passed over, being transported by FMN and iron sulfur plus the cytochromes. So after cleaving off or after oxidizing FAD, we see it loses its protons to complex 2, and this complex 2 undergoes redox reaction and it excites it. And when it becomes excited, it cleaves off these protons. And these protons cannot pass through because it's impermeable, so they have to wait to go into complex 3. Then it passes its electrons to complex 3. And this complex 3 also will become excited by undergoing oxidation reduction reactions using this electron carrier, and they pass these electrons to cytochrome C. This is cytochrome C. And this cytochrome C, after receiving these electrons are passed over to complex 4. And this complex 4 passes it over to oxygen. And oxygen is known as the last electron acceptor. Oxygen is the last electron acceptor, being moved by FMN, the iron sulfur, and cytochromes, other complex, cytochrome complex, or what we call ubiquinone. So as we see these oxidation reactions occurring, NAD giving its electrons, we see its hydrogens pass via complex 1, they can pass via complex 2, complex 3. We see other protons which are released because we cleave more than 3 hydrogen ions. We have here hydrogen ions. We see this one being moved via this complex also to the intermembrane space. We see movement of hydrogen ions into the intermembrane space via complex 1, complex 2, complex 3, and complex 4. That's why we normally say that NADH produces 3 ATPs. How? Because we see one proton being moved via complex 1, the second proton moved via complex 3, and the third proton being moved via complex 4. So these 3 protons which are produced, they come and converge at complex 5. So you find here we have 3 protons. That is for NADH. That's why we normally say it produces 3. Whereas FAD, because the FAD-H2 bypasses complex 1 and its first proton moves out via complex 3 and the second one via complex 4. So meaning for it, we see only 2 protons being moved into the intermembrane space. That's why we normally said NAD produces 3, then FAD produces 2. And what happens? After this oxygen at cytochrome oxidase, which is complex 4, accepting the last electron, it becomes reduced, whereby it will come and combine with this hydrogen. If we have 2 hydrogen here, they combine, and this combination results into formation of water. That's why we see under electron transport chain production of water, whereby this oxygen, which is the last electron acceptor, it combines with the 2 hydrogen ions in the mitochondrial matrix to form water. And oxygen, don't remember, it is the last electron acceptor. The last electron acceptor. then what happens? That these protons, remember we have been moving out protons but not bringing in protons. So there is what we have created what we call an electrochemical gradient. That these protons that have been being moved out by the electron motive force, they converge. When they accumulate, we see the intermembrane space being more positively charged and inside being negatively charged. Because we have lost protons, but we have not brought them inside. So it creates a concentration gradient. And it is these positive charges that converge at complex 5, what we call ATP synthase, that will create a force known as proton motive force. It creates a proton motive force. And this proton motive force is created by the accumulation of protons in the intermembrane space. As we know that this mitochondrial membrane is impermeable except via complex 1, 3, and 4, and they cannot come up again inside. There is a deficit inside and positivity. So this is the electrochemical gradient. That when they accumulate at complex 5, that is ATP synthase, they create a proton motive force. And it is this proton motive force that is going to come and causes the combination of ADP plus phosphate inorganic to form ATP. In the presence of ATP synthase, this proton motive force will spin, it spins this complex 5. Some books call it F1, F0, at pairs. So that this proton motive force will create a high speed, it spins this complex 5, that because of its high spinning, it will cause the combination of adenosine diphosphate plus phosphate inorganic to form ATP. And this is how ATP is formed in our body. We normally get energy by this process known as oxidative phosphorylation. This is what we call oxidative phosphorylation, whereby oxidative phosphorylation is the formation of ATP in the presence of oxygen in the mitochondria, whereby the accumulation of protons in the intermembrane space, they converge at the ATP synthase, that is complex 5, and create a proton motive force. As they are coming inside to equalize or to balance the charge, they cause a force, that proton motive force, that spins this complex 5, creating energy for combination of these two to produce ATP, and this process is known as oxidative phosphorylation. And this step, or this reaction, it was described by a man known as Peter Michel, whereby Peter Michel proposed a theory, and this theory is known as chemio-osmotic theory. It is known as chemio-osmotic theory. Chemio-osmotic theory of oxidative phosphorylation. So this is the chemio-osmotic theory of oxidative phosphorylation. And how does it state? Peter Michel stated that because of the inner mitochondrial membrane being impermeable to protons, only protons permeable to complex 1, complex 3, and 4. We see the efflux or outward movement of protons from the mitochondrial matrix into the intermembrane space. Therefore, because hydrogen ions which are positively charged accumulate into the intermembrane space, they create an electrochemical gradient, whereby here we have more positive charge than the inside. And this electrochemical gradient, that when these protons accumulate at complex 5, they create what we call a proton motive force, that when this proton motive force forces these protons to come back inside, they create energy that spins the ATP synthase, leading to the combination of ADP plus phosphate inorganic to form ATP. And that is what we call chemio-osmotic theory. What do you need to know about chemio-osmotic theory? One, that the inner mitochondrial membrane is impermeable protons. And the proton is moved via complex 1, complex 2, complex 3, and complex 4. And that movement or that efflux of protons creates a negative or a deficit inside the mitochondrial matrix and creates a positive charge in the intermembrane space. So the accumulation of these protons creates a proton motive force that when it is coming back inside via ATP synthase, it spins the ATP synthase, leading to the combination of ADP plus phosphate inorganic forming ATP. And that process is what we call oxidative phosphorylation. So that is the electron transport chain explained by the chemiosmotic theory. We are saying that the inner mitochondrial membrane is impermeable to protons, but only permeable. The proton is passed via complex 1, complex 3, and complex 4. And because we see movement only outside but not coming back, it creates an electrochemical gradient. And this electrochemical gradient, that these protons accumulate high proton concentration in the outer membrane, intermembrane space. When they accumulate, they cause a proton motive force. And this proton motive force, as they are coming back to equalize the charge, they spin the ATP synthase at a high speed, leading to combination of ADP plus phosphate inorganic forming ATP. And this is the description of oxidative phosphorylation by chemiosmotic theory. So after knowing how the electron transport chain forms ATP and water, we now need to know what are the inhibitors. What are the inhibitors of the electron transport chain? So let us look at the inhibitors of electron transport chain. And these inhibitors, I can write them here. Create space. Want to look at inhibitors of electron transport chain. What inhibits electron transport chain? We have complex 1, complex 3, complex 4, and 5. And we see complex 1, which is NAD H dehydrogenase or NAD coenzyme 2 reductase. For it, it is inhibited by a seaweed known as retinol. This retinol, the fishermen in Uganda, they use it to catch fish in lakes, whereby it is a weed that when thrown into water, it deprives the fish with oxygen and formation of ATP, and fish starts floating on water when they catch them. So this retinol, it is, for it, how do fish come on water is that they lack ATP because it is blocking complex 1. So we cannot form enough ATP. So fish float on water due to lack of energy. Retinol and sometimes the barbiturates. Barbiturates also act by blocking complex 1. You know, barbiturates like phenytoin, phenobarbital, they are given anticonvulsant patients. Patients who are they are very hyperactive. They are uncontrollable. So we can give them barbiturates and they will target block complex 1 so that they can lack energy to fight and they become calm. They become calm so that we can work on them in the hospital. Then complex 2, for it, it is inhibited by antimycin A. Complex 3, we see antimycin A inhibiting complex 3. So for it, antimycin targets complex 3. Then complex 4, we see cyanide, we see carbon monoxide, and hydrogen sulfide. And we have seen many people here in Uganda being dead of cyanide. For example, like a scenario which happened in Mbare, whereby people ate certain cassava which contained cyanide, and all of them, the 7 family, all of them died on spot. Because cyanide is a poison for complex 4. It comes and blocks complex 4, which is cytochrome oxidase, and we cannot see oxygen accepting the last electron. So still you die due to lack of energy. Even the carbon monoxide, cooking in a house with windows and doors closed, instead of forming carbon dioxide, we form carbon monoxide, and carbon monoxide can also inhibit cytochrome oxidase, which is complex 4, plus hydrogen sulfide produced by the motorbikes. Then lastly is the complex 5. This complex 5, which is ATP synthase, which is responsible for combining ADP plus phosphate in organic form ATP, is inhibited by oligomycin. Oligomycin inhibits complex 5. So in an exam, they can easily say, state the inhibitors of each complex. So you should be able as a student to know different inhibitors that we can use to inhibit these different complexes. Lastly, there are other inhibitors, and these inhibitors are known as uncouplers. We have what we call uncouplers, and these uncouplers, or you can call them uncouplers, they uncouple. As from the beginning, we have been telling you that the inner mitochondrial membrane is impermeable to the protons. So we cannot see a proton passing here, we can't see a proton passing here because it is impermeable. Only complex 1, 3, and 4. So when we are given uncouplers, we have example like 2,4-D. This 2,4-D, 2,4-dinitrophenyl, is the weed master. We use it to spray weed in our plantations. So this one, when taken, it may causes the inner mitochondrial membrane to become permeable to protons. So we see uncontrollably moving out of protons everywhere. Protons move outside and can come back inside. So we see we cannot maintain the electrochemical gradient. And without electrochemical gradient, we cannot form a proton motive force, which can cause the formation of ATP. So when you take uncouplers, no accumulation of protons outside because they freely enter in and freely move out. So it can affect, then you cannot produce ATP. That is 2,4-D, and in young children they have a brown fat which contains thermogenin, and thermogenin is also an uncoupler. So thank you so much for listening to this was electron transport chain. And this electron transport chain, we have seen different complexes, and we have also explained oxidative phosphorylation by chemiosmotic theory. Thank you for watching until the end of this video. Always remember to subscribe if you haven't subscribed, and share the video and like so that we can keep growing. Thank you so much.